Everyone. This is Dr. Pratpal Singh, an MD from CMC Ludhiana. And I had started a series of presentation on generic versus branded drugs. And in the first part of the lecture, the first series of this particular topic, I had discussed a few terms that are commonly used regarding what is the generic term, what are the act that generic drugs are regulated by, and what is an orange book. So today we'll proceed further, obviously with a recap of whatever has been told earlier, so that by the end of this session, we have some clear concept and clear view about the other terms that are used. Maybe we talk about what is a pharmaceutical equivalent, a pharmaceutical alternative, and so on, and how are acts and rules related to this particular generic versus branded drug. So let's start with our session without much delay. And we start with this session where we were talking about this generic versus branded drug. And I would start with this, with what we were talking about in the previous session. So when we talk about generic drug are identical or bioequivalent. And this is in reference to the brand name drug in dosage form, safety, strength, route of administration, quality, performance, and intended use. Now, when we talk about generics, these are well established. These are pharmaceutical products that contain well established. What they are meant to be, they are intended to be used interchangeably with original product. They are usually manufactured without a license from the original market manufacturer. They are marketed after the expiry of patent, and they are marketed either under a non-proprietor name or other approved name, or under a brand name known as branded generics. Now we are talking about a patent. So let's have a view of a patent. Now, right from the time when a new chemical entity comes into existence to the time this new drug application is approved by the FDA, it roughly takes around 12 to 14 years, and the patent rights of 20 years. So the company or the manufacturer who originally introduced this product has a patency right or an exclusivity right of maybe around 5 to 6 years, after which this particular drug can be manufactured by any other component. And based upon this, we had one of the particular landmark amendments, that is the Hatch-Waxman Amendment, for which was given in 1984. And this is considered as one of the most successful pieces of legislation. And this created the generic industry. How it happened? Well, it was 12% in 84, which increased to 44% in 2000. And this gave generic drug companies a greater access to the market for prescription drug, and gave innovator companies a greater patent life. Now, what does this act tells? The Hatch-Waxman Act says that the patent that is given to the company has the sole right to sell the drug when this patent is effective. After this exclusivity expired, manufacturer can apply to the FDA to sell generic version. And drugs, when they are submitted under this after the exclusivity, are abbreviated new drug applications where approval for the market a generic drug. The ANDA process, because why is it less costly? Because you do not require to repeat the costly animal and clinical research on ingredients and dosage forms. What you need to do is you have to go for bioequivalent and bioavailability studies, and that is why these generic medications are priced lower than the brand name medications. Now, when we compare a new drug application with an abbreviated new drug application, what you find is these three, the number 6, 7, and 8, the animal studies, the clinical studies, and the bioavailability, which form the major chunk of the cost of a drug, they are taken care by only a bioequivalence study, and that is why generic drugs are comparatively cheaper as compared to a new drug application. Now, how do we assure the quality of a generic drug? There are 5 steps of review process that are identical to the of these 5 steps which we are talking about are chemistry, manufacturing, control, labeling, testing, and the 6th step is equivalence. So bioequivalence for complicated products is discussed with the same staff that reviewed the brand product. FDA has experience with the product. So there is scientific literature that is published and the product is known to be safe. Let's talk about how a generic drug review process works. An applicant applies for the abbreviated new drug application. The application is reviewed, and if this application is found to be acceptable and complete, it undergoes plant inspection, chemistry and micro review, labeling review, and bioequivalence review. If it is not acceptable, it is sent back to the applicant. Now, if any of these processes, plant inspection, chemistry or micro review, labeling or bioequivalence are found inadequate, there is no, this particular drug is not approved. So for approval of these drugs, all these 4 passages, that is the request for plant inspection, chemistry and micro review, labeling review, and bioequivalence review have to be okay. And then an approval letter is issued. Now, the basic requirement for a generic drug is that they should have the same active ingredient, they should be used by the same route, the same dosage form, the same strength, and they should have the same condition for the use as compared to the reference listed drug. So same active ingredient, same route of administration, same dosage form, and same strength and same condition of use. Now, labeling is same as the brand name labeling, and it may delete portion of labeling protected by patent or exclusivity. And this may differ in the excipient, the pharmacokinetic data, and how it is supplied. For the chemistry part, we have the component and composition, we have the manufacturing, we have the batch formulations, we have the description of facilities, specs and specifications and test, packaging, and stability. Now, manufacturing compliance program is, the purpose of this manufacturing compliance program is to assure the quality of marketed drug product, and the product is tested by surveillance, manufacturing or testing the plant inspection, and assessing firms' compliance with good manufacturing practices or process, that is the GMP. Once this is done, this approved drug is published in the electronic Orange Book. There is an approved drug product with therapeutic equivalence evaluation. There is 23rd edition of this, which has approved and all the products that are approved by the Federal Food, Drug and Cosmetic Act. And this Orange Book, it has all the products that are approved, and there are 2 codes that are used, A and B. The therapeutic equivalence codes are A, that means that this particular drug is substitutable, and product B is that it is inequivalent or non-substitutable. And then we have the expiration dates and the reference drug lists or brand drugs identified by FDA for generic companies to be compared with the proposed. Now we talk about certain definitions that are used in bioequivalence determination. So we'll be talking about pharmaceutical equivalence, pharmaceutical alternatives, therapeutic equivalence, bioequivalence, and bioavailability. Talking about the pharmaceutical equivalent, so if a product is considered to be pharmaceutical equivalent if it has the same active ingredient, has the same dosage form, same route of administration, and identical in strength or concentration. So we are having 4 components. It should have the same active ingredient, it should be in the same dosage form, it should have the same route of administration, and it should be identical in strength or concentration. Now, there are certain other term that is used, that equivalent products contain the same amount of ingredient in the same dosage form, but this may differ in shape, release mechanism, and packaging. Then we talk about pharmaceutical alternatives. So pharmaceutical alternatives are those products that contain the same therapeutic moiety and are different salts, such as we have a typical example of erythromycin. They are esters or complex of the same moieties, are a different dosage form, or are different strengths. Now, other pharmaceutical alternatives could be different dosage forms and strength within a single product line by a single manufacturer, or it could be extended release formulation when compared with immediate or standard release formulation. Then we talk about therapeutic equivalence. So drug products are considered therapeutic equivalent if they have the following. They have a pharmaceutical equivalent, they are bioequivalent, they are approved as safe and effective, they are adequately labeled, and manufactured in compliance with current good manufacturing practice regulation. And these therapeutic equivalents are expected to have the same clinical effect and safety profile. So that is a therapeutic equivalent drug. Bioequivalence is that a drug products, they are considered bioequivalent if they are pharmaceutical equivalents, which we are talking about, and the rate of extent and absorptions is not statistically different when administered to patient or subjects at the same molar dose under similar experimental conditions. So what are we talking about? We are talking about a pharmaceutical equivalent whose rate and extent of absorption are in alignment with the parent or with the drug and at the same molar dose under similar experimental conditions. So bioequivalence could be demonstrated through in vivo or in vitro test methods, comparative clinical trials, and pharmacodynamic studies. Now, how do we go ahead with this? So there are, how do we divide this? There is a strong correlation between single dose and single dose pharmacokinetics are similar to steady state pharmacokinetics. Patients have a pharmacokinetic profile comparable to that of a healthy volunteer and 20 to 25% difference in absorption based on the maximum concentration area under have, and they are believed to have no effect, or they have believed to have similar effect on the therapeutics or no effect on the therapeutic outcome and behave in similar alignment. Coming to bioavailability, the bioavailability is the rate and extent, two terms important, two words, rate and extent to which an active ingredient in pharmaceutical equivalent becomes available at the site of drug action. So we are talking about the site of drug action. So when you talk about the site of drug action, it becomes bioavailable. Bioavailability may be assessed by measurement intended to reflect this rate and extent. Now let's talk about certain guidelines that determine bioequivalence. So generic drug manufacturing must demonstrate that a drug is bioequivalent to a reference drug product, and they need to undergo pharmacokinetic studies, pharmacodynamic studies, comparative clinical trials, and in vitro study. Talking about this particular graph, a concentration time graph, where we are talking about a study compound and a reference compound. So AUC is the area under curve, Cmax is the maximum concentration, and a difference that is greater than 20% in Cmax or AUC represents a significant difference between the study and reference compound. Tmax is time to maximum concentration. Now, how do we interpret this? Now, let's look at this. There are a reference drug that is given as a green indicator. This is the reference drug. And we have two products, product A and product B. At one time, we say product A is equivalent, while as product B is non-bioequivalent. So why do we say this? So product A is equivalent to the reference drug because its 90% confidence interval of the area under curve falls within 80% to 125% of the reference drug. We were talking about 20 to 25%. That is what is mentioned out here. In between 80 to 125%, so that this particular product is a bioequivalent. Whereas the product B, which is not bioequivalent because its 90% confidence interval is falling outside the 80% and 125% reference drug. See this? So this is product B, which is non-bioequivalent, and this is product A, which is bioequivalent because its 90% confidence interval falls within 80% to 125% of the reference drug. Now, when we talk about the pharmacokinetics regarding healthy volunteers versus patients, if two drug products perform the same in healthy volunteers, the assumption is made that they will perform the same in patients with the disease, except in the case of some drugs that are potentially toxic. So healthy volunteers and patients are considered at par for a generic drug. So I'll stop out here on the various terms that I've been talking about. I've been talking about the pharmaceutical equivalence, the pharmaceutical alternatives, the therapeutic equivalent, bioequivalent, and bioavailability. I hope I have given some clarity on this particular topic. And if there's any questions or queries, please write down in the chat box and I'll definitely get back to you. Thank you so much for your patient listening and patient hearing, and wish you happy learning and have a nice day.